everyone, and welcome to today's webinar, Using Cutting-Edge Stem Cell Technology to Model Human Cardiac Diseases. I am Sydney McNeil of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Miltenyi Biotech. To learn more, visit miltenyibiotech.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. We only have Arianne with us for 30 minutes today. Any questions that we are unable to answer will be answered by our speaker via the email address you provided at the time of registration. This webinar is educational and thus offers free continuing education credits. Please click on the continuing education window at the bottom of your screen to obtain your credits. I would like to now welcome our speaker, postdoctoral fellow, Dr. Arianne Caudal from the laboratory of Dr. Joseph Wu in the Cardiovascular Institute at Stanford University. Arianne, you may now begin your presentation.
Well, thank you very much for that very kind introduction. I'm truly honored and excited to be here, so thank you very much for the invitation. Today my talk is entitled Using Cutting-Edge Stem Cell Technology to Model Human Cardiac Diseases. So let's get started. For today's presentation, I'm very excited to share our group's contributions to the exciting field of cardiac diseases and induced pluripotent stem cell, or iPSC, technology. I'll first introduce some central themes with some relevant background, and then I'll spend a lot of time in the following three main sections. First, how we generate and maintain iPSC lines. Next, how we can differentiate these lines into cardiac cell types. And then finally, how we can use these models as viable tools to study disease, with specific emphasis on some exciting therapeutic targets that our lab has elucidated in the past few years. Finally, I'll summarize what we have learned today and discuss some possible directions for the future of this technology. So cardiovascular disease, which is broadly defined as pathologies of the heart, blood vessels, and the vascular system of the brain, accounts for nearly 1/3 of total deaths worldwide. There are various etiologies of cardiovascular disease that exist, and here I've highlighted just a few of them. But for today's talk, we'll focus on just one specific type of cardiovascular disease, which is the dilated cardiomyopathies. For the rest of the talk, this will be abbreviated as DCM. So with a prevalence of at least 1 in every 2,500 people, DCM is the most common cause of heart failure after coronary artery disease, as well as the leading indication for heart transplant. The cost of managing DCM in the US alone has been estimated to be between 4 and 10 billion annually. DCM is characterized by ventricular chamber dilation or enlargement and thinning of the heart wall, as depicted by this schematic. Highlighting just the left ventricles, DCM can be clearly observed using ultrasound, as I'm showing here from patients. And furthermore, histological analysis of patient cardiac tissues reveals very striking changes at the cellular level between healthy and DCM patients. Part of the difficulty with treating DCM is that it has really broad molecular manifestations. So mutations in genes encoding sarcomeric, cytoskeletal, mitochondrial, nuclear membrane, and even calcium metabolism proteins account for nearly 50% of DCM cases. And although there are at least 33 genes associated to date, over 75% of the known cases have mutations residing in just a handful of genes, some of which I've highlighted in this table. In today's talk, I'll further highlight two key genes, Lamin A, which structurally supports the cell's nuclear envelope, and the cardiac troponin T, which is part of the contractile machinery of the heart that helps it beat. There is a clear need for new therapies and cutting-edge therapeutics for cardiovascular diseases. However, there are still several barriers to research. So first, the cardiomyocytes are post-mitotic, meaning unlike many cell types, they will not divide in a dish, so we can't just continuously grow them in the lab. So patient samples are therefore very limited. And healthy controls, which we need for comparison, are even more difficult to obtain. So historically, animal models have been used, but they remain a low throughput and very expensive option, and it's not compatible with high throughput drug screening. Furthermore, small animal models don't fully recapitulate human cardiac physiology, and there are inherent differences in heart rate and disease progression. Our lab utilizes the induced pluripotent stem cell, or iPSC platform, to model cardiac diseases from humans. This model has many key advantages. The first is that stem cells can be differentiated into a variety of specialized cells, some of which we can recapitulate in the lab, including our favorites, the cardiac cell types, and therefore stem cells can provide a limitless material for research. In our case, this circumvents the post-mitotic nature of the adult cardiomyocyte. Second, they genetically match the people that they are derived from, meaning that we can study many different types of heart diseases across a wide range of demographics and populations, and that sample is more relevant to designing therapies. Third, the cell-based format is very compatible with large-scale screening of different drugs, gene therapies, and intervention strategies, which we can then directly test the effects of with cell-based functional readouts. So how exactly do we do this? Here is a general overview of our iPSC pipeline. We start with somatic cells from patients, usually in the form of blood or skin fibroblasts, because those are easy to extract, and we reprogram them into iPSCs. Then we differentiate into our cell type of interest, and then we use combinatorial strategies to determine disease mechanisms, then try to interrogate and rescue them. Our end goal is to find viable treatment strategies that are translational to clinical applications. So first, let's dive in detail into what reprogramming actually is and how we generate, validate, and characterize iPSC lines. I thought I would share our most recent publication from the lab, led by postdoctoral fellow Dr. Sang Cho, published in January in Stem Cell Research. Here, our lab generated 3 heterozygous Lamin A mutation carrying iPSC lines from 3 cardiac laminopathy patients. Lamin A is a rod-shaped structural scaffold of the nucleus. It forms these long tetrameric bundles that functionally support and organize chromatin, and therefore playing an important role in DNA replication and signal transduction. Mutations in Lamin A, called laminopathies, largely result in cardiac diseases, such as DCM. But the underlying mechanisms still remain unclear. Therefore, having models for laminopathies where we can test drugs are incredibly valuable resources. So from 3 DCM patients, 2 male and 1 female, blood was collected. Peripheral blood mononuclear cells, or PBMCs, were isolated from the blood using separation techniques, and then stabilized in media and supplements. Next, we use vectors based on a modified non-transmissible form of Sendai virus to safely and effectively deliver and express key genetic factors necessary for reprogramming somatic cells into iPSCs, and then clonally select for cells that were transduced. Once we have single colonies, we let the now iPS cells recover and propagate. And typically the cell culture media of iPSCs needs to be replaced every day. However, we recently switched to using StemMax iPS brew media, which only requires a media change every 2 days, which just greatly improves the day-to-day practicality of using iPSC models. And as you can see, the cells are both happy and healthy. One of the first things we validate is cell morphology and genotype. So here you can see the very quintessential stem cell morphology is present in all 3 patient lines. These patients have a mutation in the Lamin A gene, which changes guanine to adenine, it's a single base pair mutation that essentially changes arginine 133 to glutamine. And this amino acid resides in the central rod domain of the Lamin A protein. We are able to validate the mutation is present in all 3 lines using Sanger sequencing. Next, we look at the whole genome array and validate that each iPSC cell line has a normal karyotype. And then we look by quantitative RT-PCR that the Sendai virus that we use for reprogramming, which is non-integrating, is no longer present in the cell. Here we use an early passage iPSC line as a positive control and a late passage iPSC line as a negative control. And as you can see at late passages, all 3 cell lines express very low to no SEV transcript. Next, we use immunostaining to look for the key pluripotency markers, NANOG, SOX2, and OX34. As you can see, these cells express all 3 markers, which all have high detectable transcript levels. Finally, we validate each cell line's differentiation potential. So stem cells should have the capacity to become all 3 germ layers, right? The outer ectoderm, which essentially becomes skin cells, pigment cells, and neurons. The middle mesoderm, which for us is very important, right, because that yields all of the muscle. And the inner endoderm. So to confirm, we stain for germ layer specific markers and then show that these lines can therefore differentiate into all 3 germ layers and there have been successfully reprogrammed. Once we have functional iPSC lines, we then differentiate them into cardiac cell types. For today's talk, I'll focus mostly on iPS-derived cardiomyocytes. iPSCs first become committed mesoderm before becoming cardiac progenitors and then early cardiomyocytes. And as early cardiomyocytes, they will begin to physically beat in a dish. You can very easily see this by light microscope. And then further maturation of the cardiomyocytes, just with prolonged culture or other genetic or molecular interventions, increasingly starts to resemble the transcriptional profile of mammalian models. Logistically, this takes about 30 days in the lab to get from iPSC to cardiomyocyte. Starting from the iPS brew, this requires several media changes alongside interrogation of Wnt signaling to yield committed mesoderm, then addition of insulin when the cells start beating, and then 2 days of glucose-free lactate selection to increase purity. Using this strategy, we can generate patient-matched iPSC-derived cardiomyocytes. To exemplify this work, I'll briefly describe this paper from our lab, led by Dr. Ning Sun and published in 2012 in Science Translational Medicine, where patient-derived iPSC lines were used to model dilated cardiomyopathies. Here, we generated cardiomyocytes from iPSCs derived from patients in a DCM family, carrying a point mutation in the gene encoding the sarcomeric protein cardiac troponin T. Cardiac troponin T plays a role in the contractile machinery of the heart. And compared to control healthy individuals, cardiomyocytes derived from DCM patients exhibited abnormal distribution of sarcomeric alpha-actinin, shown here in red by immunostaining. And as you can see in the merge, there are these tiny puncta in the DCM cells that are not present in the control. They also have abnormal calcium ion regulation, detected here with representative line scan images and decreased spontaneous calcium transients. And furthermore, if you can take these cells and make embryoid bodies, which are three-dimensional aggregates of cells, and stress them with norepinephrine, the DCM embryoid bodies have a very poor response and greatly diminished beating rate, followed by sarcomeric disarray and cell disorganization, as you can see from the immunostaining. To summarize, the cells demonstrated in a dish common phenotypes observed in DCM patients. So, how can we further use this model to gain new therapeutic insight? So at the time, circa 2A gene therapy and metoprolol both had positive effects in DCM trials. But the underlying mechanism for why they worked was still unresolved. Hence, our lab set out to investigate if these mechanisms of DCM repair could be recapitulated in a dish. So alongside improvements in gene expression in DCM, the circa 2A overexpression improved contraction force as measured by atomic force microscopy. In parallel, they found that metoprolol treatment for 1 week significantly decreased the number of single DCM cardiomyocytes with disorganized sarcomeres. These results suggest that blockade of the beta-adrenergic pathway helped DCM iPSC-derived cardiomyocytes to resist mechanical deterioration and improve their myofilament organization. To further explore beta-adrenergic signaling in iPSC cardiomyocytes and its role in DCM, authors from our lab tried using beta agonists. So this study was led by Dr. Haodi Wu and was published in Cell Stem Cell in 2015. Here they found that the beta-adrenergic agonist isoproterenol induced mature beta-adrenergic signaling becomes compromised in disease. In order to test the effects of beta-adrenergic stimulation, iPSC cardiomyocytes were next treated with isoproterenol, and spontaneous calcium transients were analyzed. Isoproterenol increases cardiac contractility. So while the control cells respond to isoproterenol challenge, the DCM cells do not. Although expression levels of several beta-adrenergic signaling components were unaltered between the control and DCM iPSC cardiomyocytes, they actually found that the phosphodiesterases, PDE2A and 3A, were upregulated in DCM cardiomyocytes. So the phosphodiesterases essentially cleave cyclic AMP, and this is a very important process in a lot of different cell pathways. But notably, the pharmacologic inhibition of the PDE2A and PDE3A restored cyclic AMP levels and actually ameliorated the impaired beta-adrenergic signaling of DCM cardiomyocytes, suggesting that the therapeutic potential of targeting these PDEs. So these studies, especially this one, really shows, what pathways are important in the normal cell and in the disease cell, but importantly also presents a solution And that's essentially what our lab looks for in interesting projects and collaborations. In summary, I showed you how our group is using patient-derived cells for reprogramming of iPSCs. We discussed how cardiomyocytes can be differentiated. And then I showed you 2 examples of how these models have been employed to gain a deeper understanding of DCM pathogenesis. To conclude, iPSC technology offers an exceptional opportunity for creating disease-specific cellular models, investigating underlying mechanisms, and optimizing therapy. In the future, we hope to really come full circle to continue to integrate other cardiac cell types into these systems. So today I talked about cardiomyocytes. We can also make cardiac endothelial cells and fibroblasts. We can put them together in really interesting combinations and stoichiometries to really fully recapitulate that complex cellular microenvironment. And by doing that, we get closer to disease therapies that are eventually going to be more applicable to translational applications. So we want to come full circle, and we want to generate novel therapies and models that can actually help real-world patients. And to really do that in a manner that is personalized to that patient and what they need. I'd like to thank my lab and my mentor, Dr. Joseph Wu, the Stanford Biobank and CVI is listed here, and my funding sources. So thank you everyone for your attention. I'm very happy to take any comments or questions. Yeah, thank you.
All right, thank you, Arianne, for your informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you would like to ask, please do so now. Just click on the Ask a Question box located on your screen. We will answer as many of your questions as we have time for. Let's get started. Our first question is, are iPSC-derived cardiomyocytes exactly like adult cardiomyocytes? What are some limitations to the model?
That's a great question. So they are not exactly like adult cardiomyocytes. Comparatively, they are still quite immature in morphology, metabolism, and some of the ion channels. However, there has been a lot of work from a lot of different labs for strategies to improve the maturation phenotypes. So a lot of people are either using genetic strategies, changing metabolism, which means like changing the media, how much glucose to give, how much fatty acids to give. People are trying mechanical force, electrical stress, lots of different options, and a lot of them are very promising. I think to date, kind of a combined approach is the gold standard, but there are definitely ways to improve the model. So great question.
Great, thank you. Our next question is, can you put different types of iPSC-derived cells together to simulate the cardiac environment?
Yes, great question. Absolutely. I tried to kind of allude to this. This is something that I find really fascinating. So you can put cells together. You can put them as a co-culture. So in a monolayer, just in a dish, you can put cells together and see how they interact with each other physically. You can also kind of put them on different platforms, so they're not physically touching, but that's kind of a way to look at what signals are secreted between them. This allows you to kind of detect cell-to-cell communication, which is a clue to how disease propagates across the whole organ. So that's actually a very important facet of our research. The other thing you can do is to grow different cell types as three-dimensional aggregates. They're called engineered heart tissues, EHTs. You essentially take millions of cells and you grow them in a mold around two posts. And they kind of engraft and you form a tissue that beats as one unit. It's very cool. And it's much more relevant than some of the monolayer models. So yes, you can, and it's a very exciting and really beautiful part If you look in some of our publications, you can see what these look like, and I'd highly recommend it.
Perfect, thank you. Our next question is, besides genetic models, what other types of stress can iPSC-based models be used to study?
Oh, okay. Yes. So in the examples shown today, I mostly showed genetic models or stress induced by drugs, but actually we're interested in lots of different types of stress. So we're looking at kind of environmental stress. We're even looking at radiation exposure, hypoxia, changes in diet, etc. Those are all things we can certainly model on a cell-based platform. Personally, I'm also interested in physiological stress, such as exercise and pregnancy and things like that. Those are a little bit more difficult to model in cell culture, but I hope to soon get there. Thanks for your question.
Thank you. Our last question is, you mentioned using patient-derived cells, but can iPSCs be generated from healthy people too?
Yes. So this is a very interesting question. We typically look at patients just because we want to find therapies, but I actually think that you also need to look at healthy people too, right? And so from the healthy people, we're looking mostly across different ethnic groups, gender, things that can really affect the genetic background, and then trying to kind of tease out how that manifests into cellular phenotypes. It's very difficult to do that, but I think it's really an important platform to understand the cohesive model. So that's definitely of high interest here. Thank you for your question.
Thank you again, Arianne, for your time today. Do you have any final remarks?
Yeah, thank you for having me. I just want to kind of advocate for our work. If you find any part of this presentation exciting at all, please reach out. Email me with your questions. I'll get back to you as soon as I can. And also know that our lab is very open to collaboration and sharing any of these models. So please check out our lab website, the WU Lab at Stanford Cardiovascular Institute, and reach out if you want to work with us. We're very excited. Thank you.
Thank you again, Arianne, for your time today and your important research. We would also like to thank LabRoots and our sponsor, Miltenyi Biotech, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today and those submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.